Thank you. So thank you a lot for having me today. And when I say me, I mean the Paleo-ecological group, because I'm here to replace Marie-Ève Monchamp that cannot be here today. And I will try to do my best, but those are her slides, so I may not be very efficient to say them. So be indulgent, and I will try to go a bit slowly because I see that I have 20 minutes. And when I just tried it, it was much quicker. So I will try to take time. And if you have questions, you can just interrupt me. And I will try to not talk too much anyway. So yes, indeed, I will talk about unchained DNA, DNA molecules we can find in sediment, and that we could use some time to actually trace stuff from the past. And it could include human traces. And I think it's a very interesting part of it. I want to say before I start that I'm a bit out of the topic related to terrestrial diversity and tracing livestock and stuff from the catchment. I'm working with aquatic microbes like Marie-Ève. So I may have a different perspective on how to use unchained sedimentary DNA. But we will talk about that. So why we have been creating this working group in Pages named Paleo-ecological or Paleo-ecological. It's because we were all working on unchained environmental DNA research with Marie-Ève Monchamp and other colleagues I will cite later. And we thought it could be a good idea to actually go a bit out of our scope of just working within with sedimentary DNA people, and then reaching more paleoecological paleoecologists. And this is why we are here today also. And so what we did first was to establish an international unchained eDNA research network that I will describe a bit later. And in this specific working group, we want to apply this kind of approach to characterize past critical transition. And what are critical transition is when in an ecosystem you have a sum of small forcings that will cause very abrupt changes in the community or in the ecosystem. It can be a change in the trophic network from a state of a community that will never be back to what it was before. And of course, you can use many different proxies to assess such changes, not only DNA, but it's one of them, and it could actually inform on some new knowledge that we cannot get just with diatom remains or cladoceran remains. Okay. So to summarize a bit the approach, when you go into the environment, in the lake, in the forest or somewhere, you can actually pick up sample and get DNA out of that. And this is super useful because you can determine the microbial diversity of the system. But many research also shows that you can actually detect microorganisms. Fish are quite well studied now with water sample, for instance. Actually, the first study that went out with eDNA was from Francesco Fisetola, that maybe some of you know. He actually detected amphibian DNA from lake water sample. So basically, you take a liter of water, you filter that, you extract the DNA, and you can use either quantitative PCR or metabarcoding approach and many other to quantify the abundance of a specific DNA fragment or to sequence it and to identify it. And this is what you see on this specific figure. You take the sample from the different environment and you can use a sequencer to get nucleotide sequence. And here you can see you even have a mammoth because some people are able to catch mammoth DNA from sediment, but you can see other type of organism too, including plants. And what is very cool is when you have actually a sedimentary archive, you can also retrieve the DNA from this archive. And this is quite easy when you target microorganisms like plants and mammals, because you know they are not living in deep sediment. But when you target microbes, it's a bit more difficult because some microbes, bacteria and microbial eukaryotes, they can still survive in those sediment. And then it's a bit harder to actually differentiate between sediment-living microbes and the ones that come from the pelagic part of a lake or the catchment. But when you want to trace human activities, you don't need to go specifically for microbes. So this is a good thing for you. You're lucky. You can use different types of natural archives, including ice, permafrost, marine and freshwater sediment, but also soil and cave sediment. And from cave sediment, I will not talk about that later, but some very good study went out in Science and Nature over the last years from Vivian Sloan, Benjamin Verneau, Elena Zavala, that actually tracked hominid DNA from cave sediment in very different places in Europe. And those papers are crazy. They use shotgun sequencing approach, so they sequence all the DNA. They use enrichment approach, so we call that capture of a specific fragment, and then they can actually find DNA from different human lineage. We can rediscuss about that. About the overview of the approach, the sedimentary DNA approach. So as we go in aquatic system, either marine environment or lakes, we take sediment cores. So all of you know about that. From the sediment cores, you have a stratigraphy, and you can actually date sediment most of the time, and you can extract the DNA in corresponding sediment layers. Using a sequencer, you can actually authentify the DNA sequence from different organisms and use bioinformatic tools to infer past diversity in ecosystem. But of course, this is not that easy. Only the environmental DNA approach already have a lot of bias as you have to take into account. But when you take DNA from very deep sediment, very degraded, you have a lot of other methodological issues that that build up with this kind of stuff. And it includes contamination by modern DNA that is even harder to get rid of. But also DNA is fragmented in sediment, so you have shorter fragment, and then you cannot use long primer to amplify big things. So this is very technical, but this is super important. And one key thing is it's super important to have to validate the DNA signal with other paleo proxy in some specific case. Because of course it's a new field, as Emily said, and we have many stuff to calibrate still. So here, Marie-Ève put some slide to show example of why it was useful to use sedimentary DNA in the past. And you can see here a paper from tropical lake sediment, where the author actually looked at the vegetation changes over the last 50,000 years. And that's quite a long record for DNA. And they were actually able to, sorry, 5,000 years, excuse me, 5,000 years. It's possible to have 50,000 years actually, and even more. But for 5,000 years, they were able to actually track changes in plants. So it's not my specialty, but I think some of them or most of them are cultivated plant that represent stuff that human in the catchment actually cultivate in those ecosystem. And their records were validated by a complementary proxy, showing the reliability of this approach. What is very interesting also here, it's tropical lake sediment. And for sediment, for DNA to actually be preserved in sediment, usually you need cold temperature, no bioturbation at the bottom of lake water, anoxia. All of that preserve DNA better, but as you know from your study, it's also preserve the stratigraphy if you don't have any bioturbation. Here it's not the case, it's a tropical lake, but still you can catch DNA in sediment layers that are dated quite a long time ago. So it's very interesting for us. Here you can see another paper from Francesco Fisetola, 2018, where he went in Kerguelen Island in a specific place where there is a lake in the middle of the island, and he took a sediment core there with colleagues. And they actually show with DNA the presence of rabbit when the rabbit arrived on the island due to human, and how it's impacted the vegetation actually in this island. And it's very drastic. When the rabbit became very abundant, it's completely changed, provoke a regime shift or something like that in the vegetation of the island. So it's a very good proof of concept, actually, of this kind of approach. This is also a very famous paper in our field from Charlene Givekovacs from France, where she actually used sedimentary DNA to track livestock species in catchment of lakes. And I think the lakes were from France. And it is a bit hard to do that. You need to have a lot of sample, a lot of replicate to actually detect signal from mammals, because the signal is quite rare in sediment. But in this paper, she actually success, and she reproduced that in later paper. So she can see when human or actually human activity came in the catchment in the lake, and what kind of animals were brought there, and also the vegetation. I think, Charlene, if you think DNA should be included in the database you are working on, Charlene will be a very good contact for this kind of thing. And she's also one of the leader of our pages working group. But she's on parental leave now, so she has other priorities. Here, it's another paper about Lake Joux that Nathalie should know much better than me about. But what Marie wanted to show, it's the fact that it was highly impacted by human activities, so industrial activities from actually a few hundred years ago, not only the last century, but mostly the main forcing factor in this lake was eutrophication in the 80s. Like it's the case most of the in all Switzerland lake, in all Swiss lake and fresh and French lake around. Not all of them, of course, but many of the ones we are studying. And in the paper they actually published this year with Marie-Eve and colleagues, they look at different paleoproxy in the sediment to try to understand how this human activities impact the trophic network of the lake. And one of the proxy was DNA from cyanobacteria, if I'm correct, not exactly sure. Maybe it was, yeah, it was that. But they also got some Cladocera and remains from Rosalie Bruel, if I'm correct also. And all of them, they show synchronous changes related to this anthropogenic pressure. This shows how useful is sedimentary DNA to actually look for critical transition, as we call them, or regime shift. We can discuss about definition of term. We can use the term typing point also. But basically, this kind of paper shows also the complementarity of coupling paleoproxy. One of the goals of the Paleo-ecological working group is to use model to infer critical transition. And here she put something about online dynamic linear models that is very, very unknown for me. What I know is this paper from Sofia Taranu from 2018 use a model we will try to use in our work, in our upcoming work. Yes, so I finished with example about why sedimentary DNA is cool. Now I will say why sedimentary DNA is hard to use. In this specific paper we published earlier this year, we actually show all the methodological issues related to this approach. Of course, we did the review about saying, yes, it's work and it can be useful in many cases. But we have to consider that still there are stuff we don't know about. For instance, it's very hard to find fish DNA in sediment, and we don't know why. Not in superficial sediment, but in sediment are a bit old. It's a bit more harder than finding plants or stuff. We review that in this paper, and we gave a recommendation about how to do. But I hope in 5 or 10 years, someone else will publish another methodological paper going a bit forward with method, because I still need validation. It was quite fast, but the take-home message is, yes, on-chain environmental DNA is useful to reconstruct past aquatic and terrestrial diversity, and it has the potential to identify key changes related to human activity. I truly think so. In the catchment with plants and mammals, DNA, but I think also in the lake with microbial DNA preserved in sediment. Now we are more and more talking with Marie-Eve and colleagues about using this DNA to look for multi-trophic level changes It's a bit harder to catch metazoan DNA compared to microbial, but still sequencing a lot help us to get a good view on trophic network, or we hope so. And of course, we need to work a lot on the complementarity with other paleoproxy in our upcoming work with Paleo-ecological. We are interested in collaborating with other pages working group. And I think there is a clear link we have between human traces and Paleo-ecological group, and we should continue to discuss about that. And yes, I'm almost there. Yes, I don't know how much time I have left. Yeah, just want to say that we are many in the steering committee. You can see Marie-Eve here, Linda Arbrecht from Australia, Laura Epp and Kathleen from Germany, and Charlene from France. But we are even more in the, we have a support group in our Paleo-ecological group. So we try to put in front the early career researcher, Marie-Eve, me, Linda, Charlene, and keep the professor in the support group to help us doing the work. In the said eDNA we have been creating with Marie-Eve and other colleagues earlier this year, we also did this approach of the young, the postdocs, they take the lead, the postdoc and PhD, they take the lead on that, and they use the professor to do the work. And I think it's working very well this way. We are now 260 members there, and our mailing list is actually 350 members, so it's quite good. It's progressing quite fast, so we have million things of stuff to do, but we will success eventually.